Hello students. In my last lecture, I talked about Introduction to Oncology. And when I posted my video lecture in Google Classroom, one of the students asked me to explain replicative immortality and metastasis one more time because she had some confusion. So in my last lecture, I covered study of cancer, various characteristic features of cancer cells, and types of cancers. So in this lecture, I am going to talk about replicative immortality and metastasis so that I can clear any doubt that you may have. So the doubts from the previous video lecture includes replicative immortality and metastasis, which are two of the important hallmarks of cancer cells. So first of all, I'm going to explain replicative immortality. So before we go into the topic, replicative immortality, we need to understand that the normal somatic cells have limited number of cell divisions. So they can basically undergo limited number of cell divisions, and after that cell senescence happens and they die. So the bottom line is they have limited number of cell divisions. So they can divide only for a limited number of times. On the other hand, cancer cells, they can divide for unlimited number of times. So now my first question is, why somatic cells divide only for a certain number of cell divisions? And second question is, how cancer cells overcome any problem that basically helps them to divide for unlimited number of times? So we need to know that cellular aging and finite number of cell divisions for normal cells are linked to telomeres and telomerase. So normal human cells, they progressively lose telomeres. Telomere is basically piece of DNA which is present in the end of chromosome. So it progressively loses telomeres with each cell division. And consequently the telomere length reaches to a critical length, leading to a growth arrest known as replicative aging. So to understand this replicative aging more clearly, let's see the next slide. So here we can see that this is the chromosome, okay, and this blue part is telomere. So this telomere is quite important for cell division. However, during the process of cell division, when cell divides into two and DNA replicates, so what happens, a small segment of DNA within each telomere are unable to be copied. So you can see this blue part is kind of shortened down here. So this telomere is unable to be copied and are lost. So this happens because the uneven nature of DNA replication, where leading and lagging strands are not replicated symmetrically. So after certain number of divisions, so you can see, so with each cell division, this blue part is shortening. So we can see the length of telomere is shortened with each cell division. So after many cell divisions, the telomere region of DNA reaches a critical length. This is the critical length here. And at this particular critical length, cell division stops. So now we can see that how telomere length is associated with DNA replication. So in this case, we can say that cell stops dividing once telomere region of DNA shortened to a critical length. So this is the limiting factor for cell division in case of normal cells, normal somatic cells. So what happens in case of cancer cells? So in case of cancer, the replicative immortality is achieved. So replicative immortality can be defined as an unlimited potential for cellular proliferation. So now the question arises, how cancer cells overcome the problem of shortening of telomere with each cell division that we observe with normal somatic cells. So cancer cells can proliferate indefinitely, and replicative immortality is one of the hallmark of cancer cells. And this replicative immortality is achieved by overactivating telomerase. So in case of cancer cells, the expression of telomerase is very high, and the advantage cancer cells get from overactivating telomerase is maintenance of telomere length. So basically this is an enzyme which repairs and maintains the length of telomere. So that is how cancer cells acquire the property of replicative immortality. Now if we see this diagram here, so when cell division occurs, so this, let's say this orange part is the telomere, and which with each cell division, the size or the length of telomere shortens. So this leads to the aging, and after a critical point, cell division stops. So this is the case with normal cells when telomerase activity is low. But in case of cancer cells, when telomerase activity is high, so telomerase helps in maintaining the length of telomere, and that is how cancer cells can keep on dividing for unlimited number of times. So this is how cancer cells achieve or acquire the property of replicative immortality. So after this, I would like to explain the next topic, that is metastasis. So metastasis is basically the property of cancer cell to establish to other part of the tissues than the primary sites of cancer establishment. So for example, when cancer is established at a particular tissue or organ, it is called a primary cancer. So let's say if a cancer arises in lungs, so we call it lung cancer, and that is a primary cancer. But in case of metastasis, cancer cells break away from where they first formed. For example, in the above case, the primary cancer was lung cancer. So the cancer cells, they move or they break away from lungs and move to the other parts of the tissues. So they travel through the blood or lymph system and form new tumors, which we call metastatic tumors in other parts of the body. So let's say if a primary cancer was a lung cancer, and the cancer cells break away from lungs and through blood or lymph system they move to liver. So now they establish in the liver tissue. So they have moved from the primary side, that is lungs, to the secondary side, that is liver. So this property of cancer cells to break away from the site of primary cancer formation or primary cancer to the other parts of the body is called metastasis. So the metastatic tumor is the same type of cancer as the primary tumor. So even if the primary cancer was in lungs and the secondary cancer that formed in liver, the cancer still of the same type, that is lung cancer, even though it is in liver now. So this ability of cancer cells to migrate from primary sites to other parts of body is called metastasis. So in this diagram, let us say that this is cancer in intestine and this is the primary cancer. And through the bloodstream or lymph, if this cancer breaks away from intestine and establish in other parts of body, let's say liver or lung, then we call it secondary cancer, and we call this one as liver metastasis and this one as lung metastasis. So basically the primary cancer and secondary cancer, the type of cancer cell is same, even though they are in different sites. So this property of cancer cells to break away from primary establishment site, which in this case is intestine, and to move into other parts of body, for example liver and lungs, is called metastasis. So now with this, I hope that your doubts on replicative immortality and metastasis are clear. And As all of you know that Corona has become a global crisis, and during this time I urge all of my students to stay at home, and being science students I expect you to have more responsibility than others in uh, making your family members aware about corona and and about the seriousness of the problem. So stay at home and keep on studying so that your course and your academics do not get affected. Good luck, and if you have any doubt, please feel free to contact me in Google Classroom or through WhatsApp. Thank you very much. Take care.